Well, hello there. I'm back. And the last time that we got together, it was number 6. And so that makes this number 7. And what I'm going to talk about today is eukaryotic organelles. And if you remember, we ended last time with this big picture of an animal cell and with a eukaryotic animal cell with a prokaryotic bacterium right next to it so that we could distinguish them. And that was just so complicated that we said, okay, we're going to talk about that next time. So that's what I'm doing today. I'm going to talk about all the internal parts of the eukaryotic animal cell, and humans are eukaryotic animals, and so we're going to be learning about a lot about human cells today. So I've got some funnies here to get started. They're very anthropomorphic representations of what's going on inside a cell. Anthropomorphic is when we take things that are not people and make them into people, like they're doing things and acting like people, and they have quotes. So there's a cartoon on the left here where we see some of the parts. There's the nucleus that seems to be bossing everybody around because that's where the DNA is. Some mitochondria, which I mentioned before, and they have their own DNA, so they don't have to follow the directions of the nucleus. We have something called the endoplasmic reticulum, which is just outside of the nucleus, and it's doing a whole bunch of work that the nucleus is telling it to do. The Golgi apparatus, which is also working. Something called a vacuole, feeling quite fulfilled. I don't know, maybe because it's filled with water, I think. And then a lysosome, which is destroying things. So we're going to learn a little bit about these terms and these parts and what kinds of jobs that they do. The other picture that I have here on the right is kind of important in terms of the way that I think about cells. I did not make this picture, but when I think about all these things interacting and all these processes occurring, and the cell gets more complicated, but in order to do that, it has to have different parts that are doing different jobs. I think of it as a factory. And this is a nice cartoon I found of, it says a manufacturing plant. The cell is a manufacturing plant with the nucleus in the middle, and it has got DNA, and like I said, it's controlling everything. Let me see if I can't, let's see, I'll just make a circle around here. So it's controlling everything. Just outside it, we have the endoplasmic reticulum, which is where we find the ribosomes. We already know that that's where some proteins are being made, some enzymes. That's the endoplasmic reticulum, the ER. And then this over here is the Golgi apparatus, which we call the packaging plant. So we have to make things, and then we have to package them and ship them. And they might go in or out of the cell. There's lots of different things going on. But I tend to think of this as kind of a factory type of metaphor. So that's why I think this. And I stuck this into a couple of the other slides so that I can revisit it as we go along. But kind of think about all these things as interacting, doing all their own jobs, and then maintaining all the internal balance of the cell in the process, and doing all the metabolic work that it does. Okay, let's get started. All right, so this is a massive vocabulary dump here. I've already used a lot of these words, but it's a good place to come and try to review some of this material because there's no information here, just the terms, and it forces you to go, do I know what this is? Do I know what is a ribosome and why is it related to the endoplasmic reticulum? Do I know what a tight junction is and how and why it's part of the plasma membrane? What I'm going to do in this lecture is I'm going to go through each of these groups kind of one at a time. So maybe you can come back to this place and then use it as a way of making sure that we understand what's going on. So again, kind of complicated. I'm not going to go through these now because, like I said, I'm going to go through them each separately. Maybe I can dump this slide in at the end again, or I have something similar to it. So anyway, this is a good way of looking at that complicated picture of the eukaryotic animal cell and trying to see everything happening at once. This is a really good image that uses color in order to distinguish all these different parts. So not really that way in a cell. We understand that, right? Okay, good. It's kind of cartoony version here. All right, so let's go through each of these one at a time. Okay, so the first stop in that crazy slide with all the vocabulary on it is up in the upper left, and it is what's called the plasma membrane. It is the outside of the cell. It's the parts that are in contact with other cells, with the with the glop that's in the body, the fluids, and maybe other tissues. And so we have to kind of know what divides the outside environment from the inside environment in the cell. And we already know a couple things about this because we did the macromolecules and we learned about phospholipids, lipids that have a phosphate group at one end, and there is a hydrophobic and a hydrophilic end, and they form this sandwich layer on the outside. So let me put some of these things together with this first term. This is the plasma membrane. Let me just grab a color that you can see through here, okay? It's the plasma membrane, and we're taking a zoom in close look at it right here, and you can see that it's made of those phospholipids. The blue ping pong ball like structures are the phosphate parts, and these little stringy things that are hanging down inside are the fatty acids, okay, the triglycerides. And then on the other side of it, on the inside of the cell, there's an outside and an inside. On the inside of the cell, there's another phosphate. So like I said, we think about this as a sandwich, which makes it hard for things to get in or out of the cell. These pictures over here are a couple of cells. These big pink things are a couple of cells. And so this region right here is kind of what we're zooming in on. It is the membrane that divides the inner from the outer environment of the cell. And I put those in there so we can see the way that these cells connect with each other, okay? So the plasma membrane is where we started. It is on the outside of the cell. It's made of phospholipids. It controls anything coming in and out, and I don't want to give too much detail because I got a whole nother lecture about that. So the next thing in this list is, it says microvilli. There are some times that we really want to encourage the cell to exchange things with its outer environment. And the easiest way to exchange things is to have lots of surfaces that are in contact with that outer environment. So we want to increase the surface area. So instead of just being a wall, it becomes a wall that's folded and flippy floppy, because then there's lots more contact with that outer environment. And that's what these toe-looking things on this end of these cells are. Those are microvilli, tiny little projections on the outside of the cell which increase the surface area. Okay, it says here small membrane projections increase surface area and exchange. All right, the next thing on my list is junctions. These are how cells attach to other cells, but there are different ways to attach because there are different roles that a cell might, interactions that a cell might have with its neighbor. So I have three different types of junctions. That's why I moved the next three terms a little bit over to the right, because I want to talk about them separately. All right, so the first one is tight junctions. Tight junctions are permanent, fixed, and they don't let anything through. All they do is like glue two cells together, tight junctions. And the tight junctions in this picture are these little blue balls down here. And so there's one associated with each cell, and they just glue to each other. They don't let anything through. That's why they're called tight. They're not letting any material through from one cell to the other. The next type of junction is called a desmosome, and that type of junction is a little bit more dynamic. It's more movable and it's more permeable, and it can be kind of pulled apart and put back together. And so I put in parentheses next to this, it's like Velcro. Okay, Velcro would, for instance, let moisture through, but it also seals two objects together. And this middle yellow looking thing, it even looks a little bit like Velcro, is what we call a desmosome. Okay, and you see also that it's got fibers in the middle, and they are actually attached to fibers that are inside the cell. So if there needs to be something that is transported to the desmosome to be passed from one cell to another, there is kind of a conduit for that to happen. So there is some exchange. The tight junctions, no exchange. The desmosomes, okay, there might be some transport from one cell to the next. Last junction is the gap junction, and this is a place where the cells are tied together, but they are encouraging things to go back and forth from one cell to another. There is a pathway here for materials to go from this cell into the next one without leaving the internal cellular environment. It's like a doorway or a window from one cell to the next. So without having to go out into this stuff out here, which is the extracellular environment, and that can be a kind of a place where things might get destroyed or altered. Instead, it can just pass from one cell to the other. So we call it a gap junction because it's an easy way for things to be transferred, and that's the green transport mechanism right there. Again, it's proteins. All right. Just one last thing on this list, and it says cytoplasm, and that is all of this stuff inside the cell, which is kind of the watery gel material that everything in the cell is suspended in. And I included it in this because the cytoplasm and the extracellular material are exchanging through the plasma membrane and sometimes through some of these junctions. So there is because there's an exchange back and forth, it's just kind of useful to talk about it with this particular group. All right, so 1, 2, 3, 4, 5, 6, 7 terms, one of the longer ones on this group. There we go. All right then, now we're moving into the cell away from the outer surface, the outer membrane. And one of the first things we notice when we look at a picture of a cell is this big ball in the middle of it, which is the nucleus. And I have mentioned that the nucleus has DNA in it, which is kind of controlling everything. And so I've got a little list of things that are associated with that. First of all, the big purple piece here is the nucleus. And in the middle of that big purple nucleus, there's a smaller ball, which is really just a blob of DNA. And we have a name for that. It is called the nucleolus, okay? The blob of DNA, which is inside the nucleus. And then there's a boundary around that, which is called the nuclear, I'm going to say envelope. Some people say envelope. I say envelope. It's up to you. But you know that it's really just a boundary. It's not like that you're mailing something in, that envelope or envelope. But it is the boundary between the nucleus and everything else in the cell. It's kind of like the plasma membrane of the nucleus, because there is cytoplasm in the nucleus, but it has its own membrane, okay? So we have the nucleus is the big object, the boundary around it is the nuclear envelope, and the blob of DNA in the middle of it is the nucleolus. And last thing here, not too bad, the DNA is not just DNA. It is DNA plus a whole bunch of proteins that keep it organized, structural proteins that keep it in little bundles around inside the cell, or allow it to form that double helix that you've heard about. So if we take the DNA plus the proteins, we have a name for that. We call it chromatin. So it's a material that is DNA plus structural proteins, chromatin. And when we've got both of those things in there, we call all that mess, we call it chromosomes. Chromosomes are blobs of DNA and proteins that are organized within the nucleus, okay? There are also chromosomes in bacteria, but there's really only one of them, and it doesn't have to be organized because there's really only one. So chromosomes, chromatin, that's how we keep little bits of DNA separate from each other and keep them structured separately. So it's really just three things here: the nuclear envelope, the nucleolus, and the chromatin, which is how the DNA is organized in there. And that's what we have here with all these labels. We do have one other thing here, a nuclear pore, which is just an opening that goes to the outside of the nuclear envelope. And that's where the RNA is going to be leaving the nucleus in order to interact with the endoplasmic reticulum. We're going to talk more about that in later lectures, okay? So the DNA is doing something with RNA and then sending it out of the nucleus. All right, I don't think there's anything more. Yep, I got it, bye. Okay, so this is, again, we started on the plasma membrane, then we went inside of the nucleus, and we're working our way out from the nucleus, and the first thing we run into is this term I've used a couple times, the endoplasmic reticulum, or short for that is the ER. And there's a couple different types of endoplasmic reticulum. There is smooth endoplasmic reticulum and rough endoplasmic reticulum. We have those in this diagram over here. The rough endoplasmic reticulum is covered in ribosomes, these little red balls there that are basically where the RNA leaving the nucleus gets read and made into proteins, okay? So the DNA in the nucleus is the instruction book for making all the enzymes, making all the proteins. And so that information gets kind of translated into RNA and sent outside the nucleus and then made into proteins. So the rough endoplasmic reticulum is where those proteins are made, protein synthesis. And that's why we have this here, protein for export, right there. So that happens in the rough endoplasmic reticulum. Then we have this smooth endoplasmic reticulum over here, and that is where lipids are made. Not proteins, so we don't need ribosomes. We're going to make lipids over there. So rough, proteins, smooth, lipids, and the ribosomes are related to the rough endoplasmic reticulum. So I have those all right there. The next thing we have also in this diagram is the Golgi apparatus. And I mentioned before, the endoplasmic reticulum is where things are being synthesized, things are being made. Here in our factory metaphor, we call it the manufacturing plant. We're manufacturing proteins, we're manufacturing lipids, and that's why it says right here we have this big vat where we're making lipids and this big vat where we're making amino acids, where we're putting proteins together. The next place to go in our cartoon is the packaging plant, and that's the Golgi apparatus, where they're putting things in boxes to transport them somewhere. Sometimes it's to transport them out of the cell, sometimes it's to transport them somewhere else in the cell. And in the Golgi apparatus right here, that's what we see happening. We take the proteins that we're making and we send them to the Golgi apparatus, where we're going to make these little vesicles. A vesicle is a little box made out of membrane, a little blob, where we put the proteins inside and we can get them out of the cell, send them off to some other cells through the circulatory system, or we can send them somewhere else in the cell. And either one of those is considered transport. So that's one more step in the factory metaphor. Endoplasmic reticulum is the manufacturing, and Golgi apparatus is the packaging and transport. Okay, and that's what we're looking at here. Lots of things going on there, but the vesicles we'll talk about as we go forward here. All right, then next, as I move around the diagram with all the terms on it, I'm making a stop at the mitochondria, which you might remember has its own DNA and so does its own thing. Or you might remember from some class you were in where the mitochondria were called the powerhouse of the cell. And that's related to their function as well. Again, we're going to have a whole nother lecture about what's going on there. But mitochondria are basically taking the sugars, the carbohydrates that are sent to the cell to give it some energy, and making those sugars into a form of energy that the rest of the cell can use. Because the cell can't just grab some sugar and do something with it. It can't just burn it and use that energy in order to make something. It has to be changed into a form of fuel that the rest of the cells in the body can use. You can't just use sugar as a fuel, and that fuel that's going to be made is ATP, adenosine triphosphate, which we mentioned and we'll talk about a lot later on. Anyway, we have another diagram here of a big cell, so we can see the nucleus and the endoplasmic reticulum and the Golgi apparatus. No, that's the smooth endoplasmic reticulum. The green is the Golgi apparatus. See, they look a little similar. And then floating around inside this mess, we have some mitochondria. There's more than one, and there's plenty of them in animal cells because they need to get these sugars and make them into fuels. Just a few things you might notice about them to get started thinking about them is they got lots of membranes inside, lots of surface area. We learned about the microvilli where things can get exchanged. Well, these have like microvilli inside. So there's lots of areas for exchange, and we can control the flow of different chemicals, and that serves a purpose when it comes to making that ATP fuel. And I have a picture down here, which is a picture taken with an electron microscope. Zoom in way in, and we can see all of these tiny little projections that we're calling cristae, and they're not called microvilli inside the mitochondrion. They're called cristae, and they serve this function, kind of compartmentalizing the whole system. But again, this is where we're going to be the powerhouse of the cell and make some fuel that the rest of the cell can use. Okay, so again, moving around the big complicated diagram, I want to talk about something that we call the cytoskeleton. Skeleton, you know, provides structure inside the human body. This is the skeleton of the cell. That's literally what it means. Cyto, cellular, skeleton. And it's not made out of bones. It's not made out of mineralized stuff the way our skeleton is. It's made out of protein, structural protein. And there are three major types of bones in the cytoskeleton, three major types of different proteins, and I have them listed here. This is actually something that's not that visible on the big picture of the cell because it's really everywhere throughout the cell, and it would make a lot of the other stuff kind of complicated to see if we put it in the cartoon. So I have some pictures here that show how it goes all throughout the cell. So anyway, cytoskeleton provide structure and also some transport inside the cell. And there's three types: microfilaments, intermediate filaments, and microtubules. And what we're doing is we're getting larger as we go through that list. So microfilaments are the smallest of all of the cytoskeletal components. And we see them here in green, and you see how they're distributed out around that the plasma membrane, because they basically give it some structure to keep it from collapsing. It's flippy floppy, and if it needs some material to keep it slightly rigid, and these proteins provide that structure. So I've said here that it provides structure and it allows something that we call cytoplasmic streaming, which is moving things around inside the cell because there are structures that can actually glide along these filaments. They can be transported along those filaments. So microfilaments help provide that type of movement of small things throughout the cell. We'll see some pictures of cytoplasmic streaming when things are moving through the cell later on. So the next type of cytoskeletal component in my list is the intermediate filaments. They're a little bit bigger, a little bit tougher, and they also provide some structure here on the outside, but they also, you can see that they're going through the cell and providing structure also on the inside. And I've written that they provide anchorage. The reason that there's a circle in the middle here is that they're all over the outside of the nucleus, and then there are fibers that go from the nucleus to the plasma membrane, so that the nucleus is not really free to move around completely within the cell. It'll stay kind of in the middle, which makes sense because that's where the DNA is and that's where all the factory is working. Let's keep it in the middle, but it has to be anchored to something, otherwise it'll just drift around in the cytoplasm. The intermediate filaments help to prevent things from drifting completely. So microfilament, structure and streaming, intermediate filaments, structure and anchorage. The last one is microtubules. These are usually some temporary structures that are built in order to move something that's big. Move a big organelle around inside the cell, or in the case of what we're looking at here, these microtubules, it's there to move the chromosomes when the process of mitosis is taking place. A little bit of a foreshadowing here, because we're going to get a whole lecture about mitosis. You might have learned about the chromosome sliding apart when a cell is about to split, and that's what I'm talking about. Those microtubules are built in order to move those chromosomes around. I have an electron microscope picture here, which has both microtubules. They actually have a hollow center, which is why they're called tubules, and the microfilaments, and you can see the difference in size. This big one right here is a microtubule, and these smaller, skinnier ones that are creating a lot of webbing type structure, those are microfilaments. So again, kind of getting bigger as we go down the list and having slightly different functions. The last thing I have here on my list that's related to the cytoskeleton is something called a centrosome, which just means body in the middle, and it is a place, right here's one, and right here is one. It's a place where microtubules are synthesized and then digested so that they're not always there, not always in the way. So it's a place where microtubules are created or synthesized. That's what I have there. Boom, cytoskeleton. All right, so this is my last stop on the big slide with all the terms on it. This is a slide that has some words that start with V on them. You can see right here. And so that's good because maybe it makes them easier to remember, but we also have to distinguish between them. These are vacuoles and vesicles. And the first thing we need to know about them is that they are built the same way as the plasma membrane. They are built of a phospholipid bilayer, 2 layers of phospholipids. We remember that the plasma membrane is made of these 2 layers of phospholipids with a little fatty acid chains in the middle here. I'm just drawing it like a doodle, right? And here we have a picture of, I'm going to say this is a vesicle, but it could be a vacuole depending on how big it is. Where we see the same type of structure, but it's all smaller and within the cell. So it's like a tiny little cell within the cell. The nucleus is also structured in the same way. In fact, some might argue that the nucleus is a big vacuole, but here we're just kind of talking about these 2 things separately from the nucleus. So this says, it's just a little reminder, it says that on the outside of the phospholipid bilayer, we have a hydrophilic head, that's the phosphate end, and on the inside we have a hydrophobic tail, that's the lipids, and this might prevent things from moving in and out. And that's good because there's usually some crazy activity going on in this internal environment, maybe some enzymes digesting something, and then there's different processes going on out here in the cytoplasm in this external environment. But the same basic structure as the plasma membrane. The difference between them, if they're built the same, we need to distinguish them. The difference between them is vacuoles are larger and they tend to be just storing things. They tend to be just holding material that can then be released as it's needed. So a water vacuole, like in a plant cell, is holding on to a lot of water. And there are vacuoles in the animal cells that also hang on to water. They're just not as big, so we don't always call them water vacuoles. So they're holding on to metabolites or water. It's a storage area, and that's what I've written here. Vesicles are smaller and they are more dynamic. They move around on those cytoskeletal fibers, and they can grab things from the outside of the cell and bring them to where they're needed, or they can take things to the plasma membrane and release them to the outside of the cell. And we're showing that in this diagram we have here. We have a vesicle that is being formed on the inside of the plasma membrane, and it's grabbing things from the outside of the cell, and then it's going to bring them somewhere inside the cell where they're going to be used. All right, so that's internal transport. And then we have some materials that are being created on the Golgi apparatus and transported to the plasma membrane. And then when the vesicle and the plasma membrane meet, they kind of merge together so that it opens up to the outside of the cell and releases things that can be transported somewhere else in the body of the organism. So vacuoles, more static, larger, storing material. Vesicles, smaller, moving on the cytoskeletal fibers, transporting things throughout the cell. And we also see some vesicles being created from the endoplasmic reticulum, being created here from the smooth endoplasmic reticulum, and then packaged in the Golgi apparatus. We see vesicle here, vesicle here, vesicle here, vesicle here. Lots of different tasks that are going on with different vesicles. Because vesicles do so much, there are a couple that we like to distinguish from the rest of the vesicles. Lysosomes are vesicles that are doing some digestion and removing waste. So this might be a lysosome right here. And peroxisomes are digestive vesicles that are processing liquids, breaking down, not liquids, it looks like liquids, lipids, processing lipids, breaking down lipids because lipids are particularly difficult to break down. And I think that's maybe, well, this is a lysosome right here. I don't know that I have a peroxisome in this picture. Anyway, a couple of different specialized forms of vesicles. Sorry I took so long with this one, but this was the last slide in that. And I'm at 30 minutes, so I'm doing okay. All right, so I'm actually done, and this is the slide that I started with so that you can use it for review. There's nothing on this that I haven't talked about in this lecture. Now you have a lot of material to look back at and make sure that you understand everything. And I also found a couple more diagrams that I thought were great for review. One of them is very technical. It has all the language on it and is pointing to things. Actually, it's a really nice diagram. Almost everything I mentioned is in here, so it's a nice way to look back at these functions. And then there's a little cartoon here where it's very anthropomorphic. Maybe you can hear some of these organelles talking to each other. The nucleus says, "I store your genetic information." The mitochondrion says, "I produce lots of energy." I don't know what they would sound like. The chloroplast. We didn't talk about chloroplasts because those are in plant cells, and we're talking about human biology here. Lysosomes are breaking down molecules, digesting. Peroxisomes are storing and breaking down lipids. Vacuoles are storing and keeping. Again, those are less dynamic, smaller. The ER is making proteins and lipids. It's a manufacturing center. I see both the rough and smooth parts there. And the Golgi apparatus is packing proteins and sending them off. It is the packaging and shipping transport center. Anyway, nice way to review. See you in lecture number 8.